Hello, everyone. Welcome back to the Lipid Seminar Series. Guess we'll go ahead and get started right on time. I'm Amy Kiger. I'm one of the co-organizers of the seminar series now. I'd like to acknowledge ASBMB for hosting the meeting and advertising. And also the co-organizers, John Burke and Mike Arola, who initiated this great series. And now Julie Brill, Ray Blind, and Rashid Taim. So thank you for, if you've been coming before, for re-registering for our new webinar format and for these instead of the Zoom meeting format. So hopefully you'll find that not much has changed, but I would like to make a brief announcement now that we will, as before, have two talks that will change right on the half hour to our second speaker. We'll open up questions for the speakers in the chat box about halfway through each talk. And I'll call on you from the order in the chat box to raise your question then. You could pose your question yourself by speaking. But the two differences are in this webinar is that we need to ask you to please raise your hand as well so that we can locate you to unmute you. So we can't unmute everyone simultaneously. So when you type in your question, you can go ahead and just hit that raise hand button that's at the bottom of your screen. Or when we call on you, you can remember to please raise your hand so we can locate you to unmute you. The other thing is that we no longer have the ability to see your videos, so but we can hear you. So we hope that we have the same interactions and great questions and answers sections that we've been having. So if some of your friends and colleagues aren't here and aren't getting in, please remind them that they do need to re-register for the webinar link since their old Zoom meeting link won't work. All right, so with that, I think we'll go ahead and get started. So I would like to introduce Raj Rohatgi, who's at Stanford University, who will introduce our first speaker.
Thank you, Amy. Good morning, everybody, and thank you for being here. And I want to just thank the organizers for inviting us to present at this amazing seminar series. It's my real pleasure today to introduce Maia Kinnebrew, who is a graduate student in the Department of Biochemistry at Stanford. I've had the privilege of working with Maia in my lab for the past couple of years. A little bit about her. She was born in Guatemala and grew up in Southern California. She's really been a lifelong scientist. Her love of science was ignited by frequent excursions she took in the Guatemalan forest with her mother. She studied biology as an undergraduate at the University of California at Santa Barbara, and that's where she really began her interest in understanding the interactions between lipids and membrane proteins. Now, in my lab, Maia, in her courageous and ambitious way, decided to work on the longest standing project in my lab, and probably also the most challenging one, and that is our quest to understand how the hedgehog signal is transmitted across the plasma membrane. This is really a long-standing mystery in this fundamental pathway. Now, before Maia joined the project, we had converged on the idea that membrane cholesterol plays a really unexpected and instructive role in the signaling process, but there was a lot of data we didn't understand, and we didn't really have a clear picture of how the system worked. And Maia really gets the credit for putting all the pieces of the puzzle together and to come up with a really new and compelling model that explained a lot of disparate, hard to reconcile data, both from our lab and from many other labs in the field. So this highlights a final quality of Maia, which is that she loves solving puzzles of all kinds, including scientific ones. So with that, I will turn the Zoom over to Maia to tell you about her work.
Thank you, Raj, for the introduction. Can you see my screen?
Yes.
Yes, thank you. Okay, today I am excited to tell you about our recent work on the role of cholesterol in hedgehog signaling. So the hedgehog pathway is a core developmental signaling pathway that's highly conserved all the way from insects to vertebrates. And it's also important for the patterning and morphogenesis of almost every tissue in the human body. So during animal development, defects in hedgehog signaling can lead to an array of birth defects. Hedgehog signaling is also important for tissue homeostasis and regeneration. So in this context, if it goes awry, it can lead to certain types of cancers. And as Raj mentioned, a really longstanding mystery in the hedgehog field has been, how is the hedgehog signal transmitted across the plasma membrane? So unlike most receptor signaling pathways where there's one receptor that binds to extracellular ligands and then transmits the signal intracellularly, in the hedgehog pathway, this is broken into two proteins. So first we have this molecule called patched, and patched binds to the secreted sonic hedgehog ligands, and it has homology to the lysosomal cholesterol transporter MPC1. When sonic ligands bind to patched, it relieves the inhibitory effect of patched on this G protein coupled receptor called smoothend. And it's ultimately smoothend that carries the hedgehog signal across the plasma membrane. So a 30-year mystery in the field has been, how does patched actually restrict smoothend activity? And studies have shown that these two proteins don't physically interact with each other. And this has led to a model that maybe there's some mysterious small molecule second messenger that is mediating this step between patched and smoothend. And the identity of this second messenger will be the focus of my talk. So in efforts to answer this question over the years, it's led to a series of clues as to what could be going on. So the first major clue is that patched and smoothend both localize in this organelle called the primary cilia. So here I'm showing you microscopy images where the cilia is colored in red and patched is shown here in green. And you can see that patched is accumulated in the ciliary membrane. When sonic ligands are added to cells to inactivate patched, patched leaves the primary cilia membrane, and smoothend then enters and accumulates in cilia to transduce the hedgehog signal. So the fact that these two proteins localize in this organelle suggests that the regulatory step between them might occur somewhere in the primary cilia. But the reason that these proteins localize in this special compartment has remained a mystery in the field. And I'll return to this mystery at the end of my talk. So a second clue as to what could be going on is that decreasing sterol levels inhibits hedgehog signaling. And people have shown this both with pharmacological depletion of sterols using drugs like methyl beta cyclodextrin, and also in the context of genetic diseases. For example, mutating genes in the cholesterol biosynthesis pathway leads to inhibition of hedgehog signaling. A final clue comes from work from a former graduate student in our lab named Siggy Noctergale, who showed that oxygenated sterols or oxysterols can directly bind and activate smoothend. So these two kind of final clues have led us to hypothesize that maybe this second messenger between patched and smoothend could be some sort of sterol. And then in 2016, 2 lines of evidence suggested to us that this sterol could actually be cholesterol itself. So first, we are working with Christian Siebold's lab at Oxford to solve the first multi-domain crystal structure of smoothend. And so here I'm showing you that structure. And in purple here is the 7-transmembrane domain of smoothend. And smoothend also has this large extracellular cysteine-rich domain, or CRD, colored in orange. And to our surprise, there was this cholesterol molecule bound in this domain of smoothend. Around a similar time, 2 researchers in our lab, Ria Surkar and Giovanni Lucchetti, showed that treating cells with cholesterol was sufficient to activate hedgehog signaling. And using structure-guided mutagenesis from this structure, they made mutations in the hydrogen bonding network between cholesterol and smoothend. And when you made these mutations in smoothend, you no longer observed responsiveness to cholesterol. So this suggested to us that cholesterol may be activating the hedgehog pathway by directly binding to this domain on smoothend. And more importantly, these same mutations in this cholesterol binding site also blocked responsiveness of the pathway to the native sonic hedgehog ligands that inhibit patched. So this suggested to us that patched may be restricting smoothend activity by controlling ligand occupancy or cholesterol occupancy at this groove of smoothend in this cysteine-rich domain. So this is, and this data really suggested to us that maybe it's cholesterol that itself that is the second messenger between patched and smoothend, and maybe patched is regulating smoothend by controlling cholesterol. So this is where I came to the project, and I was really perplexed by this question of how can an abundant molecule like cholesterol regulate a signaling pathway that has the potential to cause birth defects in cancer? How can you regulate such an abundant molecule? So I wanted to take a step back and ask in an unbiased way using genetics, what are the lipid requirements for hedgehog signaling? So to answer this, we generated this lipid-focused CRISPR library that targets about 1,200 genes involved in anything to do with lipids, so lipid biosynthetic enzymes, carriers, transporters, and binding proteins. We introduced this library into a hedgehog reporter cell line. And then an important step for the screen was we cultured these cells in serum that had been depleted of lipoproteins. And this ensures that the cells have to be reliant on their own endogenous lipid biosynthesis machinery. And then we treated cells with different concentrations of sonic and sorted for positive and negative regulators. So here I'm showing you some of the results of that screen and kind of one way in which we represent the data. So on top here are different lipid biosynthesis pathways, and the circles below those pathways represent the genes that are important in those pathways for generating that lipid. The genes that fall below this dotted line are those that were statistically significant from our screen. And the positive regulators of the hedgehog pathway are shown in blue circles, and the negative regulators are shown in orange circles. And there were two pathways that stood out to us. The first was the cholesterol biosynthesis pathway, which were positive regulators of hedgehog signaling. And the second pathway were the sphingomyelin biosynthesis genes, which were largely negative regulators of hedgehog signaling. And a third important result that came from this is we didn't really see oxysterol biosynthesis genes as being significant hits, suggesting that it really is cholesterol and not an oxysterol that is important for hedgehog signaling. So it was nice to see our previous hypotheses kind of confirmed in this unbiased way. So now to take a closer look at these biosynthesis pathways. So here are the two biosynthesis pathways for cholesterol used in our cells. And I've colored the genes that were statistically significant from our screen in these blue circles. And what you can see is that we get both early and late steps in these pathways, which really suggested to us that it's cholesterol itself that is the relevant molecule and not some sort of precursor or intermediate. Along a similar line, here is the sphingomyelin biosynthesis pathway, and the statistically significant genes are in these colored circles. And again, we get kind of early and late steps in this pathway, which suggested to us that it's really sphingomyelin that's important. And so it was interesting that we got these two different pathways that had opposite effects on hedgehog signaling. For the cholesterol biosynthesis pathway, if you mutate these genes, you would lose hedgehog signaling. For the sphingomyelin biosynthesis pathway, mutations to these genes would lead to an activation of hedgehog signaling. So sphingomyelin hadn't really been implicated in hedgehog signaling before, so we wanted to explore this further. And to do so, we took a pharmacological approach where we use these drugs, either fumonisin B1 or myriocin, which targets this early step in sphingomyelin biosynthesis. And now I'm going to show you some data for myriocin. What we saw is when we treat cells with a dose of sonic hedgehog, we get a nice hedgehog signaling response. But if you combine this dose of sonic hedgehog with myriocin or sphingomyelin depletion, we get a massive potentiation of hedgehog signaling. And this was also true in this more physiological system of motor neuron differentiation, where typically low sonic hedgehog isn't sufficient to drive differentiation. But if you combine it with sphingomyelin depletion, we now get this dramatic differentiation phenotype. So this data showed us that sphingomyelin does negatively regulate hedgehog signaling. So why are we getting these sphingomyelin hits, and what does sphingomyelin have to do with cholesterol? So we looked into the literature and we came across these papers from Arun Radhakrishnan and his former advisor, Hardin McConnell, and they had put forth this idea that cholesterol can exist in the membrane in different pools. So first we have this cholesterol called accessible cholesterol, that's not bound to other phospholipids, so it's kind of free in the membrane. And then we have this pool of cholesterol that's bound to phospholipids like sphingomyelin, and this cholesterol we call sequestered cholesterol. And these two pools of cholesterol are really tightly linked to each other in the membrane. And you can change the amount of each type of cholesterol by changing the amount of sphingomyelin in your system. So for example, addition of sphingomyelin would favor more of these complexes, while depletion of sphingomyelin with drugs like myriosin, like I just showed you, will increase the accessible cholesterol pool. So given what I just showed you about sphingomyelin depletion activating hedgehog signaling, we thought that really maybe it was the cholesterol pool that was important for the hedgehog signaling pathway. So we wanted to directly test this. And so we worked with Arun to show that we could decrease accessible cholesterol specifically. So what I'm showing here is this protein called LRD4, which will bind to accessible cholesterol specifically in the outer leaflet of the plasma membrane, and it will trap the accessible cholesterol without changing the total cholesterol content of the membrane. And as a control, you can also use this drug, methyl beta cyclodextrin, which will strip cholesterol out of the membrane and remove it. Is when we treat with two different concentrations of LRD4 or methyl beta cyclodextrin, we would block hedgehog signaling activity. So this data showed us that accessible cholesterol is required for hedgehog signaling. And I'll remind you that I just told you data about the opposite effect, where if you increase accessible cholesterol by depleting sphingomyelin, you activate hedgehog signaling. So this led us to a conclusion that accessible cholesterol is the endogenous second messenger signaling intermediate between patched and smoothened. And maybe what patched is doing to control smoothened activity is it's depleting the membrane of accessible cholesterol to keep it away from smoothened. But this still raises an important question, which is how can patched regulate cholesterol accessibility when it's still fairly abundant in the plasma membrane? And also when this is the pool of cholesterol that is sensed by cells to maintain cellular cholesterol homeostasis. And furthermore, this accessible cholesterol in the membrane is important for a lot of other signaling pathways. So how can patched control accessible cholesterol without having unwanted crosstalk with other signaling pathways in the cell? And we thought that maybe the answer to these questions came from that first clue that I told you at the beginning of my talk, which is that these proteins localize in this special compartment called the primary cilia. Maybe by compartmentalizing patched and smoothened to the cilia, this will minimize crosstalk between patched and other signaling pathways. So we wanted to look at the distribution of these different pools of cholesterol in cilia. And to do so, we collaborated again with Arun and his lab, who had showed that you can use these protein-based probes that bind to specific pools of cholesterol. So first we have this molecule called PFO that binds specifically to accessible cholesterol. And then we have this molecule called oily A, which binds specifically to the sphingomyelin cholesterol complex. And then this useful point mutation in oily A, E69A, allows it to bind to the total sphingomyelin in the plasma membrane. So both the free sphingomyelin and the sphingomyelin cholesterol complexes. So the idea here is to take these proteins and label them with fluorophores, and then we could add them to the outside of our live cells and observe these pools of cholesterol in cilia. And so first, I just want to show you how these probes are kind of working using whole cell flow cytometry. And so here in the left panel, I'm showing you this total sphingomyelin probe. And I've done two perturbations here. So first, I've loaded cells with cholesterol, which doesn't significantly change the total sphingomyelin content. And then here I've blocked sphingomyelin biosynthesis with this drug myriosin, and you can see a nice decrease in the total sphingomyelin levels. Now, if we look at accessible cholesterol using this PFO probe, if we load cells with cholesterol or if we deplete sphingomyelin with myriosin, we see a dramatic increase in the accessible cholesterol pool. And I'll remind you that these are the two treatments that activate hedgehog signaling, both loading with cholesterol and depleting sphingomyelin both activates hedgehog signaling. And this kind of further supports our idea that it's really the accessible cholesterol pool that is important for hedgehog signaling. So now we wanted to look at primary cilia. So this is an electron micrograph of primary cilia. And it is interesting, the ciliary membrane is actually continuous with the plasma membrane. But these two membranes are kind of separated by this unique compartment called the cilia pocket, whose function is unknown. So when we stain our cells with the total sphingomyelin probe, here again, I'm showing you cilia in red. What we saw is the cilia was enriched in sphingomyelin. So you can see the cilia is lit up with this Oilier E6A probe. But if you depleted sphingolipids with myriosin, you no longer saw this staining. And what we thought is that this would, high sphingomyelin content in the cilia would likely make the excessive cholesterol content of primary cilia scarce. And so we looked at this next using this PFO probe. And indeed, what we saw is that in untreated cells, we couldn't see observable staining of the primary cilia using PFO. Only when we loaded cells with cholesterol or depleted sphingomyelin with myriosin did we see staining of this probe at the primary cilia. And so this supported the idea that the ciliary membrane has low accessible cholesterol. And this led us to hypothesize that maybe what patched is doing is it's helping to maintain this low accessible cholesterol environment at cilia, and this is how it keeps smoothened in the off state. And if this is true, then we should see an increase in accessible at cilia when we inactivate patched with sonic hedgehog ligands. So we tested. So here on the left, I'm showing you microscopy images of cilia, and on the right is just the quantification of that. So here again, cilia are in red. And in order to see staining at the cilia, we had to pre-treat our cells with myriosin. So now you can see there's some level of PFO staining at cilia in untreated cells. But when we added sonic hedgehog, we saw an increase in PFO staining at cilia by about 2 to 3-fold. And so what this is showing is that when you inactivate patched, you get an increase in accessible cholesterol in the outer leaflet of the primary cilia, supporting the idea that maybe patched is depleting accessible cholesterol at cilia to control hedgehog signaling. So this leads me to the final model, excuse me. And so here I'm showing you a zoomed in schematic of the kind of pocket membrane of the primary cilia. So I'm depicting here sequestered cholesterol, accessible cholesterol, and free sphingomyelin. And studies have shown that patched actually localizes kind of in this base or pocket region of cilia. And what it could be doing here is taking the accessible cholesterol from the cilia membrane and either transporting it extracellularly to some sort of protein or membrane acceptor, or intracellularly to some sort of protein or membrane acceptor. Simultaneously, the accessible cholesterol in cilia is kept low by the high sphingomyelin content in the outer leaflet of the cilia membrane. So these two processes together, patched pumping of accessible cholesterol and the high sphingomyelin content, ensures that smoothen stays inactive and unbound to cholesterol. Then when patched is inactivated by the addition of sonic hedgehog ligands, the accessible cholesterol levels in primary cilia rise. They may do so by kind of leaking in from the high cholesterol environment of the plasma membrane, or they could be directly delivered to the ciliary membrane by sterile transport proteins. But in either case, the high accessible cholesterol in cilia now binds to smoothened, resulting in smoothened activation and hedgehog signaling. So just to conclude, the kind of takeaways from my talk are that first, accessible cholesterol is a second messenger that communicates hedgehog signaling between patched and smoothened. Patched inhibits smoothened by depleting the cilia membrane of accessible cholesterol. And lastly, the cilia membrane has a unique lipid environment that maintains the scarcity of accessible cholesterol. And I think I'll skip this last slide and go to my acknowledgements. So I'd like to thank the people in the Rahaki Lab that made this work possible, and also our collaborators, Arun Radhakrishnan, Douglas Covey, and Christian Siebold. And with that, I would love to take any questions.
Thank you, Maya. That was a fantastic talk on a lovely paper. We do have questions coming in, so I'll hold mine for now and ask those who are posing questions to please pose it to toggle to all panelists and attendees, and also raise your hand so we can unmute you. So I'll start at the top here with Jeremy Thorner. And yes.
Can you hear me now?
Yes.
Okay, thank you. I really like the overall model. I have two related questions, however. So if you treat with myriocin, I assume you can activate the whatever reporter you use, transcriptional reporter. And my question is, in a strain where you have the D95A Y103F version of smoothened, does myriocin still activate the reporter or myriocin treatment, number one? And number two, based on our own work, and now admittedly it's yeast and there's no primary cilium in yeast, but one of the most potent ways to activate TORC2 and therefore the stimulation of the downstream AGC family kinases under its control is through depletion of sphingolipids, either myriocin treatment or just point mutations in the enzymes of the sphingolipid biosynthetic pathway. And so I'm just wondering if you checked whether or not under your conditions in your cells, there's potent activation, for example, of AKT under the conditions where you're myriocin treating, and whether or not the activation of that kinase may have some important effects on what you're seeing and may be in part of what's mediating signaling.
Yeah, to answer your first question, when we have the point mutations in the cholesterol binding site of smoothened and we treat with myriocin, we do not see any activation of the pathway. So
okay, that's good.
Yeah. And for your second question, we did not check for activation of AKT. So that may be an interesting thing that we could definitely do. We didn't look at that.
Okay, thank you. Our next question is from Gisette Reyes-Soffer. We'll unmute you if you can raise your hand.
Hello, thank you for the presentation. Very nice work. And I was interested to know about if these proteins are related to lipid rafts and how the existence or the inhibition of lipid rafts would affect the signaling.
Yeah, so I guess lipid rafts are thought to be these kind of regions that are high in sphingomyelin and cholesterol. So it's possible that when we disrupt the sphingomyelin or deplete sphingomyelin from membranes, we are disrupting these lipid rafts. And I guess that would suggest that lipid rafts are like inhibitory for hedgehog signaling, but people haven't really looked for lipid rafts or talked about the presence of them in primary cilia. Yeah.
Okay, I see a comment here that's to the panelists, but I don't know if it was meant as a question, from Amina Woods. Okay, our next question is from Dan Sleelands. Could you raise your hand so we can unmute you? Thank you.
Yeah, okay, yes. I just wondered whether you looked at glycolipids too. We get strong effects of glucosyl ceramide on MPC1 deficient cells.
We have not looked at that.
No. Okay, thank you. But I'll put my story up. I just wonder whether it's
That
related.
would be great.
Thanks
Yeah,
a
that
lot.
would be great.
Okay, we'll take another question from Bartholomew Roland. If you can read, yes, thank you for raising your hand.
Yeah, can you hear me now?
Yes.
Yeah, I was just interested in following up on the glycolipid question. You know, it looked like in your original genetic screen, you had a lot of hits in the glycosylation area. Have you looked into that any further, just broadly? I know you said you hadn't looked specifically into a lot of the glucosylceramide species, right?
Yeah, we have not. Are you seeing this screen or this here? Yeah, we haven't really looked too much into this. We have a few other people in the lab that are following up on some of these other hits here, but we haven't gotten to the so that would be definitely something interesting that we would be interested in doing.
Thank you.
Thank you.
Okay, there are more questions, so they'll be there in the chat for Maia to address, as well as we can stay after the second talk if you want more discussion. But for the sake of time, I'd like to thank Maia again for a great talk. And I will now introduce our next speaker. So my pleasure to introduce Joseph Lorent. Joseph got his PhD in cellular molecular pharmacology and analytical chemistry in Brussels, where he was studying the effects of amphiphilic substances on membranes, and also developed mass spec methods for the analysis of saponins. He went on to Ilya Leventhal's lab in Texas, where he did a number of studies, including what he's going to talk about today on the compositional and lipid order of asymmetry in cell membranes, but other interesting biophysical and bioinformatics studies of membranes. And last year, Joseph started as an assistant professor in membrane and biochemistry and biophysics at Utrecht University in the Netherlands, where he's initiating studies now on lipid oxidation and its roles in biophysical membrane properties. So thank you, Joseph, and we'll turn it over to you.
Yes, thanks, Amy. You stopped my video, actually. I'm not sure if that's necessary, but so I guess you can see my screen now.
Lori, do you know about his video? Can you fix it?
Or maybe now it works, actually.
There, okay.
Yeah, there we are. Okay, perfect. Okay, thanks, Amy. Thanks again for this nice introduction, and also to inviting me here to this meeting. So as you said already, you know, I did this work here mainly in Ilya Leventhal's lab, which was still at the University of Texas, and it was really an exciting work which we actually published this year. And so the talk will be mainly about membrane asymmetry in eukaryotic cells. So if you think about membrane asymmetry, most of you most probably think about phospholipid asymmetry, like an asymmetrical phospholipid composition in the plasma membrane, which is known since the early 70s, which was shown here by the group of Clay. And this group actually showed already, you know, that at the outer leaflet you have mostly sphingomyelin, phosphatidylcholine, and on the inner leaflet is mostly composed of PE and phosphatidylserine, which is a charged lipid. So this study was actually really interesting at this time, and they did that actually on red blood cells, and just by making an enzymatic digestion of the outer leaflet, and they combined that with thin layer or chromatography at that time. So obviously the analytical techniques, they have a little bit improved over time now. So Ilya actually and me thought it might be a great idea to basically redo the same experiment, but then with modern mass spectrometry. So that's basically what we did. We used then red blood cells from human donors. and made this enzymatic digestion of the outer leaflet. So you basically use phospholipase A2 or sphingomyelinases, and you incubate those red blood cells. You then get like a partly digestion of the outer leaflet, which then mostly transform into lysolipids or ceramides. And you can then compare the digesters versus the total membrane. And obviously you may control noise, so you're going to control if the membrane is still intact, if it keeps the asymmetry by testing for PS exposure now by NXN5. And then you're going to make this modern shotgun lipidomics of the red blood cell. And as you can see here, so this was the result. You can basically see that the outer leaflet, it corresponds pretty much to the result which was obtained in the early 70s by the Rudolf Klein with PC and sphingomyelin, mainly on the outer leaflet, and PE, PS, mainly on or almost exclusively on the inner leaflets, actually. So we actually analyzed then other species which they were not able to do so at the time, like for example the PE plasmalogen, which are like have those ether link lipids, which is here mainly on the inner leaflets, but also we have this ether lipid of PC, which is mainly on the outer leaflet. And interesting part was we also did was actually also analyzing the different acyl chains of the different lipids, because you digest them now, so you also can analyze the different acyl chains. And we found out that there's actually a big discrepancy now between compared from the inner compared to the outer leaflet, as you can see here. And if you quantify that, you can actually observe that the inner leaflet of the plasma membrane in red blood cells has actually twice as many lipid unsaturations compared to the outer leaflet. And this is mainly due to the presence of sphingomyelin on the outside. So what would be biophysical consequences of lipid asymmetry? So what would be the packing look like in a membrane basically composed of two completely different leaflets? For this, to determine this, we did first some MD simulation. And this, as you can see here, since we knew now the exact composition now, so we simplified this composition a little bit and made then two different simulations for outer and for inner leaflet. And we could really see this, this large difference, for example, for diffusion of lipids, which was much more restricted on a membrane which was simulated to be the outer leaflet compared to the inner leaflet. And also the area per lipid, which means basically the packing of the lipid, which was much higher in the outer membrane. So with this being done, we thought that's all very nice, but would it be possible to determine actually the leaflet packing or leaflet order in the different leaflets? So here that was a simulation, and it was basically like a symmetric membrane. But what about an asymmetric membrane? How would that be? Would this order be coupled? Would it be? Would you could have two different orders in two different leaflets actually? So we wanted to test that. And for this, we used here this Dye4 NAP-DHQ probe, which is a order-sensitive probe. And as you can see, for example, here, it's actually a nice probe because you can make lifetime imaging of this probe. And with this lifetime imaging, for example, you can image here GUVs, which phase separate into a liquid ordered and liquid disordered phase, where you can nicely see that the lifetime in the ordered phase is very high compared to the lifetime in the liquid disordered phase, actually. And this probe has actually another advantage. It has this double charge, which prevents it basically to flipping from one leaflet to the other. So if you incubate a cell with this Dye4 probe, we'll basically only probe the outer leaflet. And another advantage is that it is water soluble, so you can use it for microinjection. And that was actually the deciding factor because we used this probe in a microinjection experiment. And the trick which we actually used was to, for example, here you see two microinjected cells now. So one on top, one on lower. So those are actually fibroblast 3T3 cells. And the trick is to microinject this probe in the presence of BSA, because BSA has this property, you know, so that it has those hydrophobic pockets, and it can pick basically up the dye from the outer membrane, from the outer leaflet, and also the dye which has leaked, most probably through the microinjection experiment, to the outside. And then you get this nice picture here. And since you can make your lifetime imaging, you can basically mask out the plasma membrane, and there you get the lifetime, which corresponds then to your order basically of your plasma membrane. Here we did then this control experiment now where we added then the probe also from the outside here to the same cells actually, and you can actually see this very nice increase of lifetime in the plasma membrane. Which means, yes, here at this point, we basically are imaging both leaflets, the outer and the inner leaflet, and we see an increase in lifetime. And then if you only stay in the outer leaflet, we see this huge increase in lifetime. And we did then those microinjection experiments in different cells, in mast cells like RBL cells and fibroblasts. And you can see this difference between outer and inner leaflets, this order difference. And if you compare that to the GUV I showed you, you can see that the ordered phase of a GUV composed of DOPC, DPPC cholesterol with 20% cholesterol basically corresponds to the lifetime of the outer leaflet of those cells. And if you compare it to the inner leaflet, you can see that the lifetime is somehow in between. So this really means that the two leaflets have really a completely different order in cells, so completely different biophysical properties. We did then also another experiment in red blood cells because we wanted to see if in the red blood cells is the same. The problem, you cannot microinject red blood cells. So we did a little trick now. We stained from outside. And we used normal red blood cells and also used scrambled red blood cells. And we always could see that in the scrambled red blood cells, the order really dramatically decreased. Now, which also is a proof basically that a scrambled membrane, which has basically a mix of all lipids in the membrane, has a lower order than compared to the outer leaflet here. Okay, so then we know that about the plasma membrane now, but what about other cellular organelles? Would there also be like a difference in order in the cytoplasm compared to the luminal side? And this we want to test in early endosomes. So how can we do that? We can actually stain early endosomes by just incubating them with fluorescent dextran for a couple of hours. And then after this incubation, incubate them with the dye-4 probe, which is then also endocytosed and will further fuse then with the compartments which have been stained by the dextran. You can here see a cell, or a couple of cells actually, which have been incubated with this dextran. You have the dextran signal now. You can then make a contour mask of the dextran signal, which then overlaps with the dye-4 signal, and you can then determine order or the lifetime in this endosome mask. And you see here it is around 3 nanoseconds. And you can do the same in microinjected cells now, where then you have only the cytoplasmic membrane part stained, and you can see that there's definitely a large decrease in order. And if you quantify that, you can actually see that, so for a chase experiment for 15, 30, or 60 minutes, there's still always a significant difference between the luminal part, which is very higher, which corresponds basically to the plasma membrane part of the endosome, and the cytoplasmic part. So that was also very interesting because I think that was also the first time that anyone had shown that there could be some order difference in intracellular vesicles. And the question which we further asked ourselves, since we have this order difference now, would the transmembrane proteins actually adapt to those membrane properties? So what do I mean by that? For example, we made another study a couple of years ago in which we showed that that single-pass transmembrane proteins, which have a very small transmembrane domain or very slim transmembrane domain, let's say, actually prefer to partition into more ordered phases compared to transmembrane protein, which have this very large surface area of the very thick transmembrane domain, let's say. And with this, we thought, so evolutionary, or let's just say energetically, theoretically, the proteins should have the shape, like also this asymmetric shape, which corresponds basically to the packing, no? Because energetically, it's more favorable that you have this slim shape at the outer leaflet compared to this large shape. So that's why we did this bioinformatics study then. We looked at all single-pass transmembrane proteins of the human proteome and compared the exoplasmic surface area to the cytoplasmic surface area by statistically predicting it. And here we see the results. So those are all points corresponds to single-pass transmembrane plasma membrane proteins in the human proteome. And you see already that there's a symmetry, and you can see that in average, those proteins in the plasma membrane have this asymmetric shape, with exactly what we thought. The exoplasmic part smaller compared to or thinner compared to the cytoplasmic part. And this holds actually true for all eukaryotic cells or proteomes which we analyze in fungi, plants, insects. It was all the same. They all presented a asymmetric transmembrane domain shapes in the plasma membrane. And if you look closer from where this asymmetry is actually generated, you can see that in the secretory pathway, in average, the proteins seem to have like a symmetrical shape, but the asymmetry begins actually at the plasma membrane and further continues into the endosome and the lysosome. So we were then actually wondering, what's the functionality of this shape asymmetry? Could it be that this asymmetry somehow guides the localization of those transmembrane proteins? And for this, we made some artificial constructs. For example, here you see a artificial construct which is basically just composed of a transmembrane domain, and which is then bound to a fluorescent RFP. And this one here contains basically only alanines on the outside, which is a very small amino acid now. So it will create a very small surface area, and leucine on the cytosolic leaflet. So you create a very large surface area on the cytoplasmic leaflet. And you can see that a construct like this really likes to localize to the plasma membrane. Conversely, if you have an inverse shape, basically, then the protein really prefers to go to inner membranes. And we did then this experiment like a couple of times with all kinds of different constructs, and we finally found out that it was not that much the asymmetry which was responsible for this localization, but mostly the surface area of the exoplasmic part of those transmembrane proteins, where we could then see a correlation actually between exoplasmic surface area and the localization. In contrary, if we compared that to the cytoplasmic surface area, there was absolutely no correlation observable. So we thought that that was actually pretty interesting. And to summon, let's say all the results I showed you, first, I showed you that this asymmetry in the plasma membrane is first of all like a phospholipid asymmetry, now a head group asymmetry, which has been proven, also a charge asymmetry, now with charged lipids on the inside. But we also have then this unsaturation asymmetry of phospholipids, which then seems to create this packing or order asymmetry on the exoplasmic versus the cytoplasmic leaflet. And this seems to influence either by evolution, or it's a function in the cell now, which then leads to asymmetric shapes of proteins in the plasma membrane, apparently of most eukaryotic cells, actually. And it seems that yeah, this shape is also true for the endosome and the lysosome. You see it in the plasma membrane, but ER and Golgi have this asymmetric shaped proteins, which means they most probably also do not possess this order asymmetry in their membranes. Okay, and with that, I'd actually want to thank pretty much Ilya now, which was my PI over all that time in Texas, and I really enjoyed it there. It was a very amazing time we had together scientifically and even more. I also would like to thank everyone which was involved in this project, especially Lakshmi and Candice, and also Milka, which did some of the simulation. And I'd also like to thank Ed Lyman from the University of Delaware, which did those simulations experiments, and Erdinch, which actually did some results I didn't show, but he did also some measurements which actually proves that the diffusion of real proteins in the inner membrane is actually much higher than the fusion of proteins in the exoplasmic leaflet. And with that, I would be happy to answer any questions.
Thank you, Joseph. That was a great talk. Really interesting. We've got some questions piling up here in the chat, so I'll just go ahead and call on them and remind them to please hit the raise hand button so we can find your name to unmute you. So the first is from Ganesh Shelke.
Yes.
Hey, hi. Can you hear me? Yes. Thanks for the nice talk. I have just a question regarding the distribution of proteins with regards to any kind of post-translation modification that occurs to a protein. And does that affect the size of the protein and hence the distribution in the different organelles?
So your question is if, let's say, a protein which has a post-translational modification, for example, palmitoyl or myristoyl.
Yes, or any ubiquitination and things like that. So any kind of PTM that has altered size of the protein. So
that's a good question. So I did actually recently some analysis on post-translational modification, but it was more related to rats and to viruses actually. But here I didn't look for a correlation actually there, but it might be interesting to watch. Because you would think now that proteins which have a palmitoyl residue, they would prefer like a more ordered phase than.
Yeah, and targeting them to different organelles and probably degradation as well, for example, in ubiquitination.
Yeah, that's true. Yeah, you would expect it. Yeah. That's definitely a good idea. Thanks.
Great. Okay, the next question is from Clarissa Maia Montero. Looks like your talking's already permitted from before, so I think you should be able to unmute.
Yes. Can you hear me?
Yes.
Yeah. Okay. Very interesting. And I studied lipid droplets, so I was very curious to know if it would be possible to compare endoplasmic reticulum outer membrane leaflet to the lipid droplets monolayers to verify changes during formation, or if the lipid droplets can change the structure of the lipids after formation.
Yeah, that's definitely a good question. So I'm not sure if anyone tried already to investigate that. So I definitely didn't see any article about that. So it would be all speculation. So I really don't have any idea.
Yeah, interesting.
Yes. So from the ER,
monolayers.
No, that's true. So from the endoplasmic reticulum, like comparing the luminal node to the cytosolic side, it's also difficult to investigate now since it's in the cell now, so it's kind of complicated to make those analyses now. So plasma membrane is kind of a little bit easier now because it's the outside membrane. And also we did it in red blood cells mainly. Now they only have the plasma membrane, obviously.
Yeah, but then if you compare just the outer layer of the ER with the lipid droplets, then it's a bit easier because you don't need to access the inner layer.
That's true. And I mean, what could be done now, it's you could definitely use those order sensitive probes now, and then somehow trying to stain somehow selectively droplets or the ER now, because that should give you then some indications now if you microinject them or not, then at least what the cytoplasmic site looks like.
Yeah. Okay, thank you.
Okay, I see we had a raised hand early from Ed Dennis, so we'll unmute you.
My question is a beautiful presentation and talk about the asymmetry. In the initial red blood cell experiments you described, you noted that ether PE and ether PC asymmetry was similar to that of the diacyl PE and PC. Is the correlation 100% with the polar groups, that is the ethanolamine and choline, or is there any effect of ether or vinyl ether plasmalogen at the SN1 position where it's more hydrophobic compared to an acyl group in terms of asymmetry?
Oh, that's a good question. I think we never thought about that to analyzing that, but that I mean the data is there now, so we could in theory do that. So I mean, if you're interested about that, I couldn't
tell from the plot, but it looks like if you were carefully analyze that, you might see some small asymmetry.
That's true. Yeah.
The hydrophobicity. So it'd be a real test of the hydrophobicity of the SN1 chain changing.
No, that's definitely a good idea. Yeah.
Thank you.
Yeah, thanks.
Great. Okay, the next question is from Tim Levine. You also there. Okay. Can you unmute yourself? There.
Finally unmuted. Then hi again. Hi. So my question is a little bit off topic, and I think it's not a great question. It's about whether you tried this with different sterol concentrations other than the equal, or whether you ever see flipping in your molecular dynamics. Maybe it's never long enough to actually see the flipping, or even something remotely like cholesterol flipping.
Yes, so here the MD simulations have actually been done on symmetric bilayers. So you just look at the symmetrical properties. You just take the composition now from either the outside leaflet and then simulate a asymmetric one here. So we wanted to go for asymmetric simulation too. But,
I suppose it doesn't. It's not clear to me how much work it would be, but to placate the people who work on this sort of thing, maybe you could. I would suggest you could try each of their models, 80-20, 40-40, 50-50, and 20-80, and then see if it makes any difference.
Absolutely. So I think they're actually working on that right now. So I'm not sure how far they are yet, but that's definitely yeah, it's definitely interesting to know. Yeah.
Thanks.
You're welcome.
All right. Next is from Suzanne Barber.
Looks like Suzanne left.
Okay. the questions there you can get back to if you want. Next question is from Sarah Keller.
Hello, can you hear me?
Yes.
Hi, Joseph. Every time I hear your talk, it's wonderful, and every time I learn something new. So this time I really appreciated your BSA trick, and I'm wondering if it works as well in modeled GUVs. For example, does it interact with the membrane or not?
Yes, so the BSA, we did a couple of control experiments now. So actually it really nicely extracts, but only from the outer leaflet of the plasma membrane. So obviously, because otherwise you wouldn't see any more fluorescence now in the cell, which have been back extracted by the BSA, the micro-injected ones. In GUVs, it was kind of tricky because I remember that the fluorescence, it's first it's difficult somehow to measure now the intensity. And there seems to be some flipping then also of the probe, but maybe you just need the good amount of cholesterol or whatever, or really the good membrane. But we really didn't go too much into detail. But,
super, thank you.
Yeah, it might really be interesting to test actually.
Okay, next is Subrajit Banerjee. So your talking's permitted, you just need to unmute. Yeah, thanks.
Can you hear me?
Yes.
Yeah, so I was more interested in sort of the biological membrane side, and it was really cool that you tried to see ordering in some of the intracellular membranes. One of the questions I had is like, have you ever tried looking at or thought of looking at whether it works on yeast membranes? And the reason I'm asking this is because the yeast vacuole has been shown to segregate proteins into ordered and disordered domains when put under stress, especially starvation. So have you ever thought of looking into that?
Yes, it would definitely be nice, but the problem with yeast cells is a little bit the same with red blood cells. No, they're pretty small. So if you want to micro-inject those, that's kind of tough. No? So the best thing actually, which would be to have now, would be a let's say a odor-sensitive prodrug in theory now, which diffuses through the plasma membrane and then is somehow cleaved and becomes fluorescent, and then that would be something nice to have, no? But that unfortunately doesn't exist. If someone wants to try to synthesize something like this, that would definitely be nice.
Yeah, that'd be very cool.
No.
Yeah, good, thank you.
Yeah, you're welcome.
Okay, Kusum Karbanda.
Let me unmute. Thank you, Joseph. I had a couple questions. One of them you already answered regarding the lipid droplets and its stability. My other question is that we are trying to make artificial lipid droplets like perturbing the PCP ratio, which are the major phospholipids on these organelles, and using trioline as the main lipid. Can we use the probe that you're using to understand whether these changes in the PCP ratio is affecting the size and or stability of these lipid droplets? Or is the probe that you use very specific to your work?
So I guess it depends. No, the thing is, it stays at the beginning pretty much just on the plasma membrane, but then it depends a little bit on the turnover of the cell. Because it's some cells that really endocytose very rapidly and recycle the plasma membrane, basically, which means then also the probe is basically recycled and then diffuses somehow to the other organelles. Now, so I'm not sure if the lipid droplets, I mean, as long as they're connected to some other membranes like the ER, then I guess it could diffuse to them too. But I've also to caution a little bit about those experiments, because so we did some control experiments actually about the flipping of the probe. No? So it does not flip at all in the presentation, which is actually not true because it flips over time. No? So but the flipping speed is around, let's say in 1 hour you get maybe 10 or 20% of the probe which flips to the inner side. No? So that's largely enough time to make the measurements we did. But if you need to stain inner membranes, let's say selectively one leaflet, then it becomes a little bit more critical, no? Because then it depends a little bit on the turnover of the cell, how quickly the probe is endocytosed and so on.
I see. But if you're making these lipid droplets in a test tube, can we use the before?
Yeah, then that should not be a problem. Yes.
Okay, so I would email you to get details from you if that's all right.
Yeah, absolutely, no problem. Yeah, you're welcome.
Thank you, Joseph.
Okay, we have a question from Raj Rohatgi.
Beautiful talk, Joseph. I really enjoyed your paper as well. I had a couple of questions. One is, do you personally have an opinion about whether cholesterol is asymmetrically distributed and in which leaflet, since it's been a matter of debate recently? And the second question is whether you've looked at multipass transmembrane proteins and whether they show, the sort of asymmetric surface area between the inner and outer leaflets.
Yes. So yeah, it's a good question. So for the multipass, I actually did that very recently. So there's also some asymmetry, but it's much smaller now. So the thing is with multipass now, because you have a lot of area which comes together now in those complexes. Then the effect of the surrounding lipids, let's say the membrane order, does not that much influence those proteins. So you see like a slighter effect, but it's not as big. And sorry, the first question was?
The first question was just whether you had an opinion about the asymmetry of cholesterol between the two leaflets.
Oh yeah. So the cholesterol. So we did some control experiments also to look at microinjected cells versus cells stained from outside, where we depleted them then with cyclodextrin, and we looked then at the order difference before and after this cyclodextrin extraction, and we could actually see that both order of outer and inner leaflet is actually decreased. So I guess we could maybe use those experiments if we would look at the kinetics of the extraction, maybe even at the asymmetry, but yeah, we didn't go that far. So but it is certainly on both leaflets, but how much, if it is more on the outer leaflet, it's really difficult to say. But I guess there are a couple of studies which point into that direction. So there's most probably a little asymmetry of cholesterol, I would say, even if the flipping speed is really high. It's difficult to tell.
Sure. No, thank you.
Controversial subject.
Yeah, thank you. That's
our last question for Joseph will be from Skylar Budd, which is kind of along the line of what I wanted to know as well too. So Skylar will.
Hey, so I was just curious if you've done any testing on how membrane curvature has an impact, like if you tested smaller and larger cells, how the unsaturation difference changes, and if that has an impact on transmembrane proteins.
That's a good question. So we did not look at the distribution over the curvature, because you get that much signal now. It becomes difficult to separate. I mean, if you would have an idea how to test. So we did though test, let's say like the order difference in let's say in small unilamellar vesicles compared to large unilamellar vesicles, and then test those probes, and there we could definitely see a difference between let's say outer leaflet order compared to inner. So there's definitely an effect on curvature on the order now. So then looking at the proteins, that's definitely a good question if the protein shape is different. So I did it actually. I analyzed viruses also of different curvature and sizes. I looked there more at palmitolation. I could actually see that the higher the curvature was, the more those proteins had palmitol residues. But I looked also at the asymmetry, but I can't remember if the effect was that big or not. But that's something I definitely could look up. Yeah.
Yeah.
I think it was
Good
not
talk.
that significant. Okay, thanks.
Okay, great. I'd like to thank everybody for participating, attending, and especially our speakers for 2 awesome talks. Thank you, Maya and Joseph.